Now that we've covered the rod-shaped bacteria, let's go to the circular bacteria, or cocci, that are gram-positive, low G plus C. So the first one would be Streptococcus, and the example organism for this one would be Streptococcus pyogenes, and this is the most common cause of strep throat. So that would be an example of a species of Streptococcus causing problems for humans. Now, on the positive side, there are species of Streptococcus, not S. pyogenes, but there are species of Streptococcus that, like their cousins, the lactobacilli, produce lactic acid during fermentation, and we use them in the food industry. We use them to make yogurt and some cheeses. So, like the other genuses, Streptococcus has species that benefit humans and species that do not benefit humans. Now that we've talked about cocci that form chains, or the Streptococci, we're going to talk about the Staphylococci, which from their name you can tell that they form grape-like clusters. Now I've got two examples of Staphylococcus. The first one is Staph aureus. Staph aureus is a common inhabitant of human mucosa, usually on the inside of the nose, in the throat, likes a little wetter environment, but it can be found on the human skin. Usually it causes no problems, but it is one of the most serious opportunistic pathogens that we have, because sometimes they can pick up toxins either through plasmids. That would be an example of conjugation or transformation if the plasmid happens to be floating around free in the environment, or through phages. And if they pick up these toxins, then they can cause some serious diseases. Also, Streptococcus aureus, some strains of it have picked up multiple antibiotic resistance. And the most serious one is methicillin-resistant Staph aureus, or MRSA. You may have heard of that one. So we keep an eye on Staph aureus. Now the second example is Staphylococcus epidermidis, as you can tell from the name. It is the most commonly isolated bacterium from human skin, and it usually doesn't cause opportunistic infections, unlike Staph aureus. Usually, Staphylococcus aureus, when it's on the skin, is kind of a placeholder. So from this picture, which was, this is not off of human skin, that's not human skin, but imagine that in this sim, this is human skin, and you say this is all Staph aureus, then when a real pathogen comes along, it has a hard time finding a place to grab a hold, because the places are being taken up by Staph epidermidis. So it's good to have it on your skin. Now, it can cause opportunistic infections, especially on implants. Say I'm going in for a knee surgery, I'm getting an artificial knee, aseptic technique was not followed during surgery, and some of my skin bacteria get on to what's supposed to be a sterile artificial knee. Well, Staph epidermidis grows really well on metal surfaces, especially inside the body, and they form biofilm that is very hard to treat with antibiotics. In fact, they're so hard to treat with antibiotics that with artificial knees, artificial hips, when these biofilms develop, oftentimes they have to go in, take the implant out, put another one in. Eeh. So if any of you go into any surgical type applications, make sure that you're really, really serious about aseptic technique so that this doesn't happen to your patients. Now on to the mycoplasmas. Remember, mycoplasmas are most closely related to Clostridia. As far as relatedness, I should have talked about them right after the Clostridia, but they're easiest to cover and I remember them best after the strep and staphylococci. Mycoplasmas originally were classified as gram-negative bacteria, and they stain gram-negative because they don't have a cell wall. Somewhere along the line, even though they're more closely related to Clostridia, which are gram-positive bacteria, mutation or mutations occurred that they lost the ability to produce peptidoglycan. Because they don't have a cell wall, they are pleomorphic, as you can see from this beautiful sim. See, some of them even have kind of a funky Y shape. And some of them form cocci, and some are rods, and it's because they don't have a cell wall to keep them in one shape. You're probably wondering, well, if they don't have a cell wall, how do they keep from popping in a hypotonic solution, which most bacteria experience at one time or another? Well, mycoplasmas have two answers to this. One, they form a very tough cellular membrane. It's reinforced with steroids. Now, bacteria usually don't make steroids. Most bacteria do not include steroids in their cell membranes, but this helps reinforce the cell membrane of mycoplasmas. They don't make their own steroids. In fact, when we grow them in culture, and these are some beautiful fried egg colonies, at least that's what we call them because that's what they look like. If we grow them in the lab, we have to add cholesterol. So fried egg, adding cholesterol, get it? Anyway, bad joke. And so you're probably wondering, well, do they only grow on eggs? Well, actually, mycoplasmas are parasites, at least the ones that we've been able to isolate so far. That's the other way that they get around the hypotonic problem, is they live inside of other organisms, multicellular organisms, and we pretty well take care of the salt concentration problem for them. And they have access to cholesterol and other steroids, and they cause disease. So I have two examples. Mycoplasma pneumoniae. It is the most common cause of atypical pneumonia, or what we commonly call walking pneumonia, where you feel like you got pneumonia, but you don't feel so bad that you stay flat on your back in bed. And then there's M. genitalium, which causes urinary tract infections. So I can't think of any mycoplasmas that benefit humanity. Also, if you get them in your tissue culture when you're trying to grow viruses, they're really a pain because, being pleomorphic, they can wiggle through filters, and it's very annoying. They're, so far as I know, the only bacterium that can get through filters. Well, that's it for the low G+C gram-positive bacteria. Now on to the high G+C gram-positive bacteria. And we're going to cover, under this category, Corynebacterium, Mycobacterium, and Actinomyces. Now, this is as good a time as any to mention Mycobacterium and Mycoplasmas are easily confused because they both start with myco. So remember, these guys are bacterium, the other ones are mycoplasmas, even though they're bacteria, because they have that reinforced plasma membrane. That's how I keep them separate. First, the Corynebacterium. Now, Corynebacterium are rod-shaped. If you look at them under a transmission scanning electron micrograph, sometimes they form these kind of funky oval shapes right before they divide. Now, Corynebacterium are distinguished by an unusual cell division method. So you see here we've got a cell that has divided. It's got a cell wall separating the two daughter cells, but instead of just separating in binary fission, they undergo what's called snapping division, where basically it kind of snaps and a hinge remains so that they're kind of connected by a little strip of cell wall. And this causes them to form V-shaped structures like you see here, and palisade arrangements where they line up longwise, as if they had divided this way instead of this way. These palisade arrangements and V-shape are characteristic of Corynebacterium. So when you see these under the microscope and they're gram-positive rods, you pretty well know that you've got Corynebacterium. The example of Corynebacterium that I'm going to use is Corynebacterium diphtheriae. This is the causative agent of the disease diphtheria. Diphtheria used to be a very severe childhood illness, and what happens is you get a pseudomembrane across the esophagus, which leads to difficulty in breathing, and oftentimes the children die. Now, it can still be a severe childhood illness, but we have a very good vaccine for it. Next up in the high G+C gram-positive bacteria are the Mycobacterium. Now, Mycobacterium are rods, but they're in the acid-fast group. In other words, they produce mycolic acid, a wax, that they embed in their peptidoglycan cell wall. So they are the first example of our acid-fast bacterium. And this is a micrograph of an acid-fast stain. Now, there are non-pathogenic Mycobacterium, common soil inhabitants. They're involved in turnover of dead leaves and other organic molecules, making them available for other organisms. But the most famous ones are pathogens, of course. The first one is Mycobacterium tuberculosis, and this is the causative agent of TB, tuberculosis. And they cause chronic disease. They get into the lungs. By the way, they're aerobes, so it makes sense that they would like the lungs. Lots of air coming in and out. And they get in, and because of their mycolic acid in their cell walls, makes them very hard for white blood cells to phagocytize. Because of the mycolic acid, they grow slowly, which is another reason why they cause chronic illness. Mycobacterium leprae is the causative agent of leprosy. Thank goodness we pretty well have that one under control, because it is not a pleasant disease. Next, on to the actinomycetes. Originally, actinomycetes were thought to be fungi. They're filamentous, they live in the soil, and sometimes they put up aerial shoots, if you want to think of that look an awful lot like fungal fruiting bodies. But they have no nuclei, so they got kicked out of the fungi. Also, they have peptidoglycan in their cell walls, so they are bacteria. But we still have in the name the fact that they look like fungi. As soil bacteria, they are involved in the turnover of carbon within the soil. They break down dead material, and they also produce antibiotics. So here are three genera of the actinomycetes. There's the one that we named the group after, Actinomyces. We have a few opportunistic pathogens with them, not very many. Common soil bacteria usually doesn't cause people problems. Nocardia, which is responsible for the smell that we get, that we associate with fresh turned dirt. And Streptomyces. Streptomyces is a genus that we have isolated several antibiotics from, including streptomycin. Well, that's it for the gram-positive bacteria. Next, we're going to cover the gram-negative bacteria, and then we will be done with the prokaryotes.